So let us start with today's episode of blood grouping and blood transfusion. Now, what are the learning outcomes of today's episode? So as I have told you that it is a very short topic, we will be only discussing about the blood groups and the method how the blood transfusion is done depending on the blood groups. So let us start. Now, in the year 1901, Karl Landsteiner, he was a German biochemist, he first said that the blood of different individuals has some biochemical differences. Now it was found because when blood was transfused to one another, few people after blood transfusion, several complications started in their body, and it was fatal also at times. So he did an experiment to see whether the composition of blood of each and every person is same or not, but he found that there are several biochemical differences. So what are those biochemical differences? Now our human blood consists of protein, two types of protein in human blood. So one is called agglutinogen and another is agglutinin. So what is this agglutinogen? This agglutinogen is the antigen. It is a glycoprotein which is present on the surface of RBC. Now, if you look my pointer here, suppose this is the RBC of the blood, and these A, what are written on the top, they are some glycoproteins. So this is blood group A, and it is having agglutinogen A. Is it clear? So it is also called the corpuscle factor. So it is a glycoprotein which is present on the surface of the RBC, and it is present in every blood group type, but it is different in all. Now suppose in type A blood group we are having A agglutinin, whereas in type B blood group we are having B agglutinin. In type AB we are having both A and B agglutinogen, but in type O, there are no agglutinogen present. That is, no antigen is present on the surface of RBC of individuals having blood group O. So we will learn about it in details in the next slides. What are they called? It is an exception in blood group type that it is not having any antigens on its surface. So rather we can say that these all are certain exceptions which is present. In AB we can see both the antigens are present, and whereas in O no antigen is present. Now let us come to the second protein factor, that is agglutinin or antibody, which is it is also a protein present in blood plasma. It was present on the surface of RBC. It is the second one is present in the blood plasma and is called the plasma factor. So just dear students, remember it. The previous one, or the agglutinogen, is called the corpuscle factor, whereas the agglutinin is called the plasma factor. Now there are two kinds of antigen, A and B, and two kinds of antibodies, A and B, in blood. So the first two are present in the corpuscle or on the RBC, and the last two, or the antibodies, are present in the blood. Now let us move on to this diagram and see what is happening. Now here the blood with the antigen A is having antibody B. Type B blood having antigen B will have antibody A. Now type AB, which is having antigen A and B, will not have any antibody. So these are the biochemical differences. And blood group having type O is having antibody both A and B. So why it is so? It is the difference, or the biochemical difference in our blood is like that only, and depending on the presence of antigen and antibody, the process of blood transfusion is done. So let us proceed with the next video. Now it is clear to all of us now that what all antigens are present in the blood groups. So those all groups together are called ABO blood grouping. Okay, it is depending on the antigen present on the surface of the RBC. As given below, we are telling that ABO blood grouping system. So when group A, how we will tell? That is with antigen A and antibody B. It is always dependent on the agglutinogen or the antigen. We will not describe the blood group as the antibody is present. So we will not tell that this blood group is blood group B because antibody is B. No, it's incorrect. So what is correct? We will tell it will be depending on the group, it will be depending on the antigen present rather. So group A contains antigen A and antibody B. So have a very clear picture of this one. So it will be very easy for you to move on with the further lessons. Now group B with antigen B and antibody A. Group AB with both A and B antigens but no antibody. And whereas group O has no antigen but has both antibodies A and B. Now depending on this only our blood transfusion happens. So how does it happen? That we will see. Let us move on to the next one. Now here let us start with a pictorial representation of ABO blood group system. Now we have taken here group A, group B, group AB and group O. So let us first take blood group A, which is having antigens A. So if the structure of antigen A is like this, it should be present on the surface of RBC as we have discussed earlier, if you can recall from the second or third slide. Now it is having antibody B, and let us come to blood group B having antigen B all over its surface, antigen B, this is antigen B structure, and having antibody A. Whereas blood group AB is having both antigen A and B. So these are the structures of antigen A and B which is present all over the surface of the RBC, and no antibodies are present here. Whereas when we come to blood group O, there are no antigens present on the surface, and so antigens is none, and antibody present is anti-A and anti-B. That is, antibodies present are A and B, both are present. So have a clear idea about this blood groups and the antigens and the antibodies what are present. Then it will be very clear for you for the next slide, that is on blood transfusion. Let us move on to that. Now here let us see, this is a slide on blood transfusion. So now knowledge about the blood groups is obviously essential for the safe blood transfusion. Or what else will happen? The antigens of the donor's blood will react with the antibodies of the recipient's blood. Suppose you are giving blood group A to a person having blood group B. So what will happen there? The antigens of donor's blood, that is antigens of A, will be reacting with antigen B, and they will be clumping of RBC. Thus antigen A present in the RBCs of blood group A reacts with the antibodies of the plasma present in the blood group B of that individual, and vice versa will happen. So then what is happening there? That process is called agglutination. So what is agglutination? That is clumping of the RBC when there is a wrong transfusion of blood. That is, a person having blood group A if given blood group B, or vice versa. So they will be reacting with the antibodies of the present in the plasma and with the antigen. So an agglutination is a process which leads to various consequences. Serious consequences may happen, and which may even prove fatal. Fatal means that is life-threatening. So what we should do? This is the blood group chart we know. So blood transfusion should be done accordingly. The person having blood type AB is having antigens A and B. Antibodies, no antibodies. So can give blood to only AB, but they can receive blood from blood group AB, A, B and O. Why? Because there are no antibodies to react with these all antigens. So blood group AB is called the universal recipient. So the person having blood group AB is called the universal recipient, as it is not having any antibodies, and the antigens of all these blood groups will not have any antibody to clump with it. That is, agglutination reaction will not happen. So it will take blood from all the blood groups. Now next let us move on to blood group A. They can, having antigens A, antibodies B, can give blood to A as well as AB, and can receive blood from A and O. Okay, now why O? We will learn it in that last part, because O can give blood to everyone because it is not having any antigen. So antigen is nil, so no reactions. So blood group O is always called the universal donor. So it can give blood to all the blood groups A, B, AB as well as O. Now what happens in case of antigens? Now blood group B, what is it doing? It is giving blood, it is having antigen B, it is having antibody A. It can give blood to B as well as AB, because we have seen here that AB can take blood, receive blood from all. Is it clear? So it is receiving blood from all. So it can take blood from having blood group B also, and it can give blood, it can receive blood from B and O, and it can give blood to B and AB. Now last but not the least comes the blood group O. So this blood group O, having no antigens, having antibodies A and B, can give blood to AB, A, B and O. That is all the blood groups in the ABO blood group system. It can give blood to, and it can receive blood only from O. So it's the universal donor. As AB can receive blood from all, it is called the universal recipient. As there is absence of antibodies, so it can receive blood from all, no agglutination reaction. Here also, as it is not having any antigens, but it is having antibodies, it can give blood to all, but it cannot take blood from all. It can take blood from only O. So let us move on to the next slide and see. Now, as we have discussed in the previous video also, I think it is quite clear. Just here I have written a few words regarding that. So RBCs of blood group O, they lack antigens and are not clumped by the antibodies present in the serum of the recipient's blood. So it means blood group O can be given to persons with blood group O, A, AB and A. So hence the persons with blood group O are called universal donors, and the same thing like the persons with blood group AB, they lack antibodies in their plasma, and so they can receive blood from A, B, O and AB blood groups. So such persons are called universal recipients. So this is what I told in the previous, while telling or discussing about in the previous chart. I think this part is very small, very short, and I have tried my best to teach you in a very easy manner. So go through it, and in the next topic I'll be back with a small part again, that is the rhesus factor. In one part only I'll be discussing that. So I could have taken this in this part also, but it is becoming quite a vast episode. So next part will be only discussion about the rhesus factor. So please go through it. Don't forget to subscribe to my channel so that you keep on getting the updates, and stay updated whenever I am posting my videos. So don't forget to write your queries, and don't forget to like and share. So as of now, goodbye to you all, and waiting for another new episode. Thank you.